Hello, well, last session, so good job on the stamina. You may be wondering why I'm up here again for a completely different topic. Well, my background is atmospheric science, which means anything that moves or flows through the atmosphere I get extremely excited about, hence the smoke, wildfire, and on this side, moth sex pheromones and beetle aggregation pheromones. So what I'm going to talk to you about is a series of work that we've done, which the Forest Growers Levy Trust has supported. And it started way back in 2015, where we had an MBIE-funded program, and it was basically about developing tools for eradication in the urban environment. We called that the Urban Toolkit. That ended in 2018 with some exciting results, and so we put a rebid in trying to get that program continued. MB came back saying it was a fundable proposal, but outside their portfolio. They did say our science excellence should improve if we're going to rebid. So we did that. We teamed up with PFR. We asked them, hey, according to our stakeholders, which is a whole range of industry from forestry to vintage to kiwi fruit to MPI, they're all in the room with us. They want us to go after a $4 million bid. Will you fund that? And they said yes, as long as it's justified. What do we get back? Too big. So that was a little crushing. But in the meantime, I want to thank the Forest Growers Levy Trust, because in this interim year, you have helped fund some of these projects to continue on and continue that momentum. Now, what I want to talk to you about are three projects that we worked on during this year. Two were an enhancement of new discoveries using the projects from that Urban Toolkit, that original program. The first was further refining our lightweight mobile electrointegrogram technology. That is a lot of words. So we're going to call that the PAEAG, or cyborg, which you may have heard about. We also looked at spray application efficiency from multi-rotor UAV, so unmanned aerial vehicle spraying systems, and that's for the targeted protection area. We also started a new program to get a baseline idea of what might be happening out there, because if you think about pathogens, there's very little ways we can detect a pathogen before we see it expressed on the host plant. And by then, it's usually too late. So our idea was, what if we can get some instrumentation out there that actually detects pathogens before the host plant tells us it's there? And we were looking at ways that volatile organic compounds are emitted from plants. And that was the new project that we started in this interim year. The eventual goal here is that we would have a similar lightweight system that we developed for insects, which is the cyborg that you see in the lower corner. Laser doesn't work, all right. So here, I first want to talk to you about our portable electroantennogram, or our cyborg. It's not really a cyborg in the sense, in the strictest technological nerdy definition, where the organism is hooked up to the technology and it can move things, but we're going to call it the cyborg for short because it's close. So the background is that many insects use chemical odors such as pheromones to communicate to each other. If you're a moth, the female puts out a sex pheromone to bring in the male. If you're beetles, you send out a pheromone that says, hey, come to this tree, let's attack it so we can overcome its defenses. If you're a parasitoid, you look for the pheromone that you want to, of the insect that you want to infect. So if you have a system where you can attach that insect that senses that pheromone, so in this case, we know that females are calling with a specific pheromone, sex pheromone, to the males. If you take the male and put the male inside a system that's attached electrically to their antenna, you could actually see them discover that pheromone, and then you could follow that pheromone plume to the female, find the female. This is important in an eradication sense, because if we get a moth that comes in that we don't know the synthetic chemical for, so the synthetic pheromones, we don't know how to attract them, we don't know how to trap them, we can't find them, we can't know where that population is, we need the actual live moth to tell us where that population is. So that's the background there. Now, the idea is that we took something from about this big, which was a bench, and previously in the forest, there's been some about this big, very heavy, that were called mobile, and we shrunk them, and we wanted to get it on a UAV. And what you see here is version 4.3. It's very lightweight. It's just under 0.6 kilograms. It's highly flexible. And the project that we did in this interim year with the funding was to continue to develop this cyborg, to test it in different scenarios. So the Canadian Forest Service contributed significant in-kind funding, and we took it over to their lab, and I'll show you some pictures. We worked with it in a wind tunnel, and we took it out into the field, and we worked with gypsy moth. So gypsy moth was our targeted boogeyman species that we wanted to make sure we really knew how to use this system for. So I just want to show you some pictures of what it looks like. On the left, you see a moth, the gypsy moth. It's sort of the pretty antenna there. It's restrained inside a cup. Now, we made this cup so it was flexible to take any size of insect. So it's adaptable. So we could have a small beetle all the way up to a very large beetle, big antenna, for example. We made the unit light and strong, so it's very robust. You could carry it around and drop it. It's not going to break it. We also put on an integrated global positioning system, so we got the whole thing lightweight enough that we could start adding some gadgets like the GPS, which is highly important. So now the thing can send back basically GPS locations of where it's seen the hits of the pheromone. Now, we took it initially into a 30-meeting testing facility in Canada, and what you're seeing here is the whole sort of wind tunnel approach. You can see hanging down, that's the fan. So these big fans are blowing air. And way back in the back, we have the pheromone source, which is just a synthetic pheromone source. And then we have our lightweight unit with a live moth inside it attached. And what we're doing is we're seeing inside this turbulent environment that we can control, can we still see a signal of pheromone, 30 meters away. So we know we could do it in the lab. We know we could do it up close. Now we're introducing turbulence. So here's just another picture of what it looks like. You could see the device mounted, and that moth is inside, and then down at the very end, you could see the pheromone source. And we were getting significant hits way down here. In fact, it was so good, the Canadian researchers got excited and borrowed it for 2 weeks and looked at it with the spruce budworm. So then we wanted to go outside, and it's important to be able to take it outside. And the first step to go outside is just a grass field. There's little turbulence, but there's intermittent wind. It's a calm day. We have to make sure we know where that source is, even though the wind could be blowing from all locations. And so what you see here is you see the setup. We have a wind vane set up, so we know the wind direction. We can backtrack the plume. And way down there at the very end is our source. We tested it at 60, 100, and 130 meters away. And what I'm going to show you is just preliminary results of responses of the insect in the field. And so the pheromone source was 100 meters away, and what you see there is sort of the general noise that kind of always exists coming off the antenna. And then every so often we have the big peak and then drop, and then a big peak and then drop. Now what we haven't done, and the reason why we're saying that this is preliminary, is we don't know 100% for sure that this is pheromone because we have to go back to our wind data and back calculate the wind direction, which is standard atmospheric science and easy to do. So that is our next step, and that's ongoing. But it's pretty exciting because we're pretty sure that that's pheromone. So what are the next steps? So we need to improve our data processing, and this is actually what's holding the whole thing back from being a true cyborg. One field of data, one field day of data collection equals 3 days of processing. That is not right. So we need to get some machine learning in here. We need to reduce this time, and this will automate the process. And then we could actually put that code into the whole device, and then it's on the device, and the device can self-turn itself. We want to continue field testing. The more we work with this, the more we work with different species that we want to make sure we know how to detect, the more ready we are for, if there is an incursion, to actually take it and use it during an incursion. So I want to talk to you about one of the new programs we initiate, and this is coming after that one because this is the one where we want to build a device just like that, but for detecting pathogens before they express on their host plants. So the background here is all plants emit characteristic volatile organic compounds, VOCs. You could smell them. So you go into the pine forest and you smell it, that's VOCs. Now affected plants emit a different suite of VOCs compared to healthy plants. So that's known research. So our project was actually to look for those VOC biomarkers that are telltale of pathogens interacting with the host. So what's unknown is it specific plant pathogen biomarker, right? Or is it just sort of this general mix? But first, we have to determine if the pathogens themselves emit VOCs. And so our questions were, we looked at the Phytophthora species, and our questions were, do different Phytophthora emit different VOCs such that you could identify them just based on the VOCs? And so what was done is there were some samples from the headspace of cultures. So in the bottom of that tube, there's a culture of the Phytophthora, and it's closed. So it's emitting whatever it's emitting, it's respiring, and then we draw the gas out and we run it through the machine. And we tested this on 3 different Phytophthora species, the Kari dieback, the red needlecast, and Cornoviae. So then the next step after this sort of experiment is to see are these emissions identifiable if they're inside the host plant? And so the plan was to invest cardley seedlings with this and then test it. That didn't work. The Kari diebacks did not take into the seedlings, but I can show you what we did with the headspace. Now, what the take-home message from this is on the left, we have the Kari dieback, that's all green. The red needlecast is red, and the Cornoviae is blue. And what we're seeing there is there's very specific signature, especially for the dieback, but also the red needlecast can be parsed out. This is pretty exciting. So the next steps are to put this inside live plants and see if we could still see this signature coming out live plants. Now, I just want to touch base on the last project that was supported through this program, which was targeted control with unmanned aerial vehicles. Now, SCION has a very large program in this space. We look at things in the field, so doing spraying applications in the field. We're looking in the lab, and we're using wind tunnels and computational fluid dynamics, so the big, hefty computers. We've done all sorts of investigation of the wake, of how low to the ground, how fast can it go. Spot spraying for wilding control is underway, and flow of air, characterizing the flow of air is also underway. But just as a reminder, there's really good practical applications here. It's for urban pest eradication, wilding conifer control, weed control and boundaries, all sorts of different applications. But you have to know what you're spraying and where is it going. You can't just put it out there and hope that it lands there, right? So we need to look at how is it depositing. Now, what we have found is that there's a significant amount of spray moving away from the swath. So the swath is sort of the paintbrush stroke of the UAV, especially for those small droplets. And that's not really surprising. That happens coming out of helicopters as well. But there's no significant effect of droplet size on that shape of the swath. So the swath has greater concentration in the middle and coming out, and that is kind of interesting. And that means that probably turbulence is dominating that pattern. Now, we tested everything with an Agras MG1, and lane separation in a low, lightweight, light wind crosswind was 4 meters. So that means you could take your UAV every 4 meters and assume you're getting some decent coverage. So methods for estimating spray accountancy and spray material are robust. And the reason I said that is because UAVs, there's lots of spraying drones out there, but no one's actually really gone back and done the science behind it. So we first had to develop the methods for estimating the spray and where is the spray going. And we really have to do that because you have to account for the driftable fraction, what's going off your border, what's going into the riparian, and in the eradication programs, we want to make sure we get maximum efficacy onto the ground. So what are our next steps? We will continue to work in this space, and I wanted to highlight some work that we're doing at the University of Canterbury through a PhD, which is to characterize the flow of air and spray below and between rotors. The greenish image is in the lab. It's actually live spray. There's a propeller running, and there are computers tracking the aerosol droplets. And on the right is the model simulation. So this is a hefty computer. 1 hour real time is about 4 hours computation time. And it's determining the individual flow of those. Now, why is this important? It's important to know where this spray is going. Is it going up? Is it going down? Could it be driftable? But it's also important for some of these sensors. So if we want to mount our cyborg, which is going to be sucking in air and looking for a pheromone, or we want to suck in air and look for pathogen signatures, we have to know what that airflow looks like and where it's coming from. And we have to characterize what that UAV is doing. If you think about helicopters and fixed wing, years and years of research went in there, especially from the military, to characterize that airflow that we've leaned on. Hardly any research has done it in the UAV space, and that's why SCION has taken on this large program. Perfect timing. Thank you.